Thank you very much, Richard, for that kind introduction, and sorry that I can't be there in person today. I hope you can see all the slides okay. So yeah, I shall make a start now. So a brief confession to make to begin with. When I gave my title to Corinne and Josie some months ago, I wasn't quite sure what I was going to talk about. So I gave quite an anodyne title, thinking it would open up a lot of sort of possibilities. And so now I have sort of an alternative title to reflect what I want to speak about today. So the new title is All Catalysts Are Equal, but some catalysts are more equal than others. Now, this isn't just my sort of love of George Orwell novels where I got this quote from, but it relates to a conference talk I saw a couple of months ago. So I was at the Operando 7 conference in Switzerland, and I saw this really fascinating talk about machine learning and how we can design improved catalysts. The paper's not published yet, but the preprint's available, and there's a link here on this slide. But this group could really improve performance for very complex multi-element composites through using generalized descriptors to improve catalyst performance. So these descriptors include electronegativity, they include binding energy, and
slides are not moving.
The slides not moving.
Just on the first slide.
Okay. You still on what slide are you on? The title slide.
The title slide.
You can see the PowerPoint, but it's not in presenter mode.
Okay.
It's online, Richard.
Okay, just give me one moment. Apologies all. Right.
So we can see the presenter view rather than you need to swap the display.
I know, I'm not a Zoom, unfortunately I'm a Teams person, so I'm trying to work out how to do it. So
on your PowerPoint, if you click display settings at the top, so if you go up now, slide your mouse up.
Unfortunately, the Zoom bar is hiding the PowerPoint. Okay, display settings, now I've got it. Okay, is that working now?
Yes.
Okay, awfully sorry about that hiccup. Hopefully we're there now. So what I was sort of saying was, I was at this talk in Switzerland and I was sort of really impressed by this sort of work on machine learning to make new materials. So using generalized descriptors like electronegativity, like binding energy, previous catalyst performance, they could really improve and design multi-catalyst sort of multi-element compositions. And it was really quite impressive. And again, the preprint for the work is here. But it made me think actually that they were making these very complex multi-phase materials, and without knowing anything about how the catalyst was made, they could actually end up designing better catalysts. But in our experience, the catalyst preparation really matters. So I thought it would be quite good today just to take you through some examples we've done with the hub over the last decade to really talk about why catalyst preparation matters and how that might be important for the future. Okay. So the first example I'd like to talk about is some work that Ellie Dan did. She was a UK Catalysis Hub PhD student. She finished sort of several years ago now, but Ellie was sort of co-supervised by myself, by Alex Gogay, and also Chris Hardacre at Manchester. So it's a real collaboration, and that's what the Hub's been really great for. And one of the first bits of work that Ellie did was to look at the effect of different precursors on how you can improve your particle size distribution. So here we have some TM images of different palladium on alumina particles that are prepared by wetness impregnation. The first material is a palladium nitrate impregnation salt, and the second material is a tetraamine palladium hydroxide impregnation salt. And what you can see is they follow the same procedure. You impregnate with your precursor solution, and then you calcine at 500 degrees for 2 hours, and you can see these different sort of distributions. But what we wanted to do was try to understand why the different precursors gave rise to these sort of different particle size distributions. Why did the tetraamine palladium hydroxide yield smaller particles? And this coincided with some combined techniques we were setting up at Harwell sort of at that time on combined X-ray absorption and infrared spectroscopy. So when we think about that process of going from a molecular precursor all the way to a supported particle, then you can use IR to look at the functional groups of the ligands adsorbed to your precursor, and you can use X-ray absorption spectroscopy to follow the metal speciation and that sort of genesis of nanoparticles. So we thought this would be a really good method. And you can see on the screen here, there's sort of two setups we have now. Initially, we have the conventional Herrick sort of dome approach that allowed us to take IR and ZAFs. And now we've moved and progressed that to a spatially resolved system where we can actually look across a fixed bed profile at different axial positions and measure ZAFs and IR at the same time. But this work was done on the initial case of the single point Herrick system. So let's look at the X-ray absorption data of the tetraamine palladium hydroxide precursor on the alumina support. And I appreciate most of you probably aren't XAFS experts, but the key thing to focus on there is the chi data. That's the oscillatory structure as a function of reciprocal angstrom. And you see it's a very sinusoidal pattern, and the intensity is mostly towards low K, which basically means probably light neighbors and limited long-range ordering. When we look at the impregnated precursor, so palladium nitrate on alumina, this is before the any calcination, what you can see is that the pattern is broadly similar for the chi data, but you begin around K9, where there's this sort of red asterisk, to actually get a more complex pattern. And this complex pattern arises because you're starting to see palladium-palladium scattering interactions. And that's manifested quite well when we look at the Fourier transform of the XAFS data. So here we can see our Fourier transform. It's a pseudo-radial distribution function. So the features at a certain distance give you an indication on where you might expect to see a scatterer. And we can see we can fit this contribution in our XAFS data from this palladium-palladium interaction. So even before we've calcined our sample, there's already a localized recognition of where the palladium sites are, so that they're close by even before we do a thermal treatment. And that's really important when you think about particle aggregation and making larger particles. Now here we have the calcinations done with the X-ray absorption as a function of temperature. And what's clear here is when you look at the palladium nitrate sort of calcination, you can see you begin to get these features around 3 in 3 angstroms in your Fourier transform, which is an indication that you're starting to form a palladium oxide phase, and this is sort of increasing in size. In terms of the amplitude of these features with respect to the palladium oxide reference, because you're working at high temperature, you'll never get exactly the same amplitude because there's some thermal disorder which dampens the signal, but we are moving towards a bulk palladium oxide structure. Interestingly, when you look at the tetraamine palladium hydroxide, when you the calcinations this material, it's not until about 400 you really start to see features indicative of palladium oxide. So you're really restricting the growth of your particles by using this this precursor. And then finally, to finish this sort of little bit of the story off, we have the drifts data. And what the drifts data showed us was that actually, because you have nitrate as a ligand, it's able to bridge between different palladium sites, and it's actually coalescing these palladium units. And that's quite important for them making these larger aggregates earlier. But interestingly, for the tetraamine palladium hydroxide, we can see that the bands associated with the amine groups are actually lost around sort of 150. But that doesn't correspond to the point where we begin to get a bit mobile palladium atoms or growth of palladium oxide. And actually you can see there's some adsorbed nitrosyl bands that persist on the surface for quite a long time. And only when they're removed, we begin to see the sort of growth of the particles. So these adsorbed nitrosyls might actually limit the migration of the palladium atoms on the lunar support. So that was quite a nice example of, not just that sort of ingrained knowledge of, why, which precursors give rise to sort of better particle size distributions, but what actually causes it. And of course, one thing that I think is really important to say is that in terms of our supported nanoparticles, you may have a structure to begin with, but during catalysis, these nanoparticles are very dynamic. So with Emma Gibson, we did some work on gold palladium nanoparticle restructuring. Again, this was a really early hub project, almost 10 years ago now. And here, this was really nice where we could use our ZAS and our drift to actually see how gold palladium particles initially had some palladium oxide on the surface before CO oxidation, but during the CO oxidation reaction, actually, we ended up with a sort of more core shell type structure where your palladium goes to the surface and you end up with a very gold-rich core. So yeah, using your ZAS and IR, you can really learn a lot about how nanoparticles evolve by making your catalyst and also during catalysis itself. So changing tact a bit here, we're going to look at another way we can make catalysts, which is through mechanochemistry. And this was a PhD project of Rachel Blackmore. She was funded by Johnson Matthey, and she was based full-time at the Harwell campus. And this is an example of, that importance of that sort of Harwell research center. And so in terms of mechanochemistry, it offers a really interesting route to make materials and produces far less solvent waste. So there's quite a drive in the industry to do this. So in Rachel's work, we were looking at planetary ball milling. So here we can see a picture of that setup of the actual planetary motion and the individual jars and how they rotate. And within each jar, a series of milling media and your catalyst. So in Rachel's work, what Rachel was looking at was the production of perovskite catalysts, specifically lanthanum manganese oxide. And so on this slide here, what we have are two bits of data. We have the XRD composition analysis as a function of time of milling, and also the X-ray absorption data as a function of time of milling. And in terms of what was being milled, Rachel was starting with pure phase oxide, so manganese oxide and lanthanum oxide. What's interesting when you look at these data is that actually they sort of tell you a slightly different picture. The diffraction tells you that between 3 and 4 hours, you have 100% phase pure lanthanum manganese oxide. But the XAFS does not tell you that actually. It tells you something slightly different. So here we can see that the Sol-Gel reference samples at the top of your Fourier transform data there. And you can see, yes, from about 2 hours, your XAFS data begins to resemble that pure phase material, that it never reaches the same pattern. So whatever you have, it's not a completely pure crystalline lanthanum manganese oxide. So the sample is, that you prepare through mechanochemically is sort of structurally more diverse. And so when we do some other analysis, when we do some XPS analysis and some TM with EDX, what we find is that when we look at our milled materials, we have this peak that's higher intensity for the oxygen 1s XPS, which is linked to higher adsorption of oxygen-containing materials linked to oxygen vacancies. And also we can see a greater degree of oxygen vacancies from the EDX analysis towards the surface. And that's very important for the catalysis here. So Rachel looked at the activity of these materials towards N2O decomposition. And what we saw is when we looked at the samples prepared by mechanochemistry, that the onset of N2O decomposition was much lower in temperature. So the performance overall was quite comparable to the Sol gel, but this earlier onset of decomposition, we could really link to those sort of oxygen vacancies, which you get by making your sort of slightly amorphous form of the lanthanum manganese oxide. So we've spoken now about making supporting nanoparticles through impregnation, by making bulk oxide phases through mechanochemistry. And so now we're coming back to look at making nanoparticles, but now by Sol immobilisation. And this was a collaboration of mine that started many years ago at the research complex with a good friend and colleague of mine, Professor Nikos Dimitratos, now at the University of Bologna. And we initially started on this work together with a PhD student in collaboration with Richard, Dr. Scott Rogers, and then recently George Tierney has also taken this work over and completed his PhD as well. So we were really interested to learn more about the systematic steps we could change in our sort of Sol mobilization process to make a supported particle, whether it's the choice of surfactant or the temperature of reduction or many different parameters. But the work that we want to focus on with Scott initially was how the reduction temperature might change your nanoparticles, and also the solvent you use for that reduction step. I mean, Scott came to us with this idea, and essentially there's two ways this could work. If you go to higher reduction temperature, you might have faster nucleation of your sort of precursor and make your particles, and that could give you a smaller particle size. Or by working at higher temperature, you could accelerate the growth phase and also, get larger particles. So it was quite interesting the result we were going to get. But what Scott actually found was when we work at lower temperatures, we were able to make smaller nanoparticles. And the way that's manifested in the data here, so here we have excess data of gold and titanium particles prepared with a PVA and sort of water solvent at temperature of 1 to 75 degrees. And in terms of the Fourier transform for the excess data, the magnitude of those features you see between 2 to 3 angstroms, they are associated with gold-gold scattering distances. And as the intensity of that feature drops, that means your coordination number is dropping and by inferential particle size is dropping. So if you work at lower temperature, you can achieve smaller particles. So we thought, hey, that's great. We have a really neat route to making controlled smaller particle sizes. So obviously low temperature is great for doing that. So let's change the solvent system and let's do the work in ethanol water mixtures. And again, the same thing, we can go to lower temperatures now because we aren't limited by the freezing point of water. And again, we can get really nice small materials by this preparation route. But what's quite interesting for these materials was when we looked at their performance for glycerol oxidation, there were particles of the same sizes prepared by both the water and water ethanol routes. So by TM, the same average particle size diameter, but actually when it came to their turnover frequency, completely different, almost an order of magnitude apart. And this was quite fascinating, actually. So the selectivity profile was broadly similar. They did essentially the same thing. There was no difference in selectivity, but activity was completely different. When you looked at the gold on titanium particles that were prepared through the water-only route, so what we saw when we looked at CO adsorption with drifts is that the adsorption bands were very different. In the region around 1900 wave numbers, where you typically see bridge bonded CO on gold surfaces, in the series prepared through water ethanol, you typically get very broad bands, and that's sort of indicative of this sort of dipole coupling you get between all your different sort of bridge COs absorbed on the surface. But we saw very, very defined bands of adsorbed CO at these sort of at these specific sites. And so that was sort of indicative of, actually not just sort of, larger planes of CO, but clusters. And then here when we did some sort of TM, what we saw, we could actually identify significant populations of atoms and clusters. And actually we link the performance not just to the average particle size, but to the presence of these cluster species. And again, knowing the different preparation routes is quite important to understand, and how you can make that inference. So that was work from Scott Rogers. And so then with George Tierney, we try to follow that by looking at these materials, in solution on the beamline. So what we wanted to try to understand was the importance of the immobilisation step. And we really wanted to assess the colloid solutions at the concentration we typically make them at, which is very low concentration. So I think we typically around 50 micromolar. And that's a real challenge. I mean, trying to get excess data on such dilute systems is very difficult. But I-20 scanning at diamond light source is an absolutely amazing beamline. If you want to work at dilute concentration, it's a really good place to go to. And so here we made our colloid solution. We then flowed it through a sample cell, and we measured our excess data in fluorescence, and we made sure that there was sort of no change over time. And we also made sure there was no fouling on the reactor or the windows of the reactor. And what this did was it allowed us to decouple the properties of the colloid in solution at different concentrations and the properties of the colloid once it was immobilized onto a titanium support. And what we found was that we could change the concentration of the colloid in solution from 50 to 1,000 micromolar, and the excess data in terms of statistically was essentially identical. There was no change. So you can work at all these concentrations in solution, the particle size is exactly the same. But interestingly, it was when you then immobilized that higher concentration solution onto the titania, then you saw the particle growth. So here you begin to recognize that immobilization part of making your catalyst is really important on how you control the growth. And maybe there's ways you can work at higher concentrations and produce less solvent waste. So this was work from George Tierney. And so now with I-20, the fact that we could work at such dilute concentrations, what we thought would be an absolutely fantastic experiment was to actually follow the birth of these nanoparticles under realistic conditions at those dilute concentrations. And this is exceedingly difficult. There's papers in the literature that try this with SACs, and they typically do it at much higher concentrations, but we wanted to use X-ray absorption. We really wanted to look at that nucleation phase, not just the growth phase of the particles. And so we developed this sort of flow setup, which is a coaxial flow. So essentially you have this sort of T-junction here on the screen, and through the center of one of the outlets, you have your gold precursor. Then on the outside of that, you have a flow of your reductant and your capping agent. You also need to make sure you add some fluorinated oil and some things to suppress hydrogen production. And George was fantastic in really being diligent in reducing the fouling, so we could be absolutely certain there was no fouling of the gold precursor on the wall of the Kapton tube that was our reactor. And we could then follow this at the beamline. So obviously you spend a lot of time designing all these experiments, but there can always be things that you forget. So this is sort of an example of, we're almost there, there's a little bit more to do. So we took the equipment to the beamline, but we saw lots of oscillations in our data. And this was basically the pulsing of the syringe pump. So at these concentrations, that just the pulsing of the syringe pump was enough to give us this noise in our signal. So we were able to mitigate that on the experiment. And here we can see, as a function of the position from the inlet towards the outlet, we can see the change, the precursor. So position 1's right at the the part where you have the mix in, and then further along the reactor profile, you can see you're getting reduction of your gold chloride, you're going towards sort of metal particles. So this setup works, providing we can get the right pump, which pumps which do exist, And this will actually enable us to follow, those very early stages of nucleation and actually get EXAFS data and try and understand the structural events that are taking place. So it's not complete success there, but we definitely made really good strides to understand those initial steps of nanoparticle formation through colloidal methods. So on this sort of same topic, Scott also used this method for palladium nanoparticles. And again, different solvent systems, different temperatures. And this time it was palladium on titania, and we're looking at furfural hydrogenation, specifically trying to understand what gives rise to different selectivity in terms of furfural alcohol and tetrahydrofurfural alcohol. And the results here were really curious. So again, the A series of catalyst is prepared in water. The B series of catalyst is prepared in water ethanol mixtures. Increasing number is essentially higher temperature. And what we see here is that as we increase the particle size in the water system, we get, by high temperature of colloidal reduction, we get increased selectivity towards furfural alcohol and decreased selectivity to tetrahydrofurfural alcohol. But interestingly, although the particle size is going in the same direction, in the water ethanol system, you get the inverse trend. You begin to get reduced selectivity towards the furfural alcohol, and you get higher selectivity towards tetrahydrofurfural alcohol. So essentially the same catalyst, palladium on titania, but inverse particle size effects in terms of selectivity. And we thought this was fascinating. When you look very carefully at the availability of surface sites by looking at CO adsorbed on the palladium and titania surface, what you see is that the ratio between linear to bridge CO changes on these different systems. There's a far greater proportion of bridged CO for the water ethanol series, which favor the complete hydrogenation product. But when you're looking at the water series, it's the opposite. So actually, it was the availability of these surface sites which dictated the overall selectivity of the catalyst. And we propose or hypothesize that by changing the choice of solvent system, it's changing the interaction between the surfactant and the metal nanoparticle. So George took this a step further and screened different linear alcohols and found that methanol was the best alcohol in terms of making the smallest particle size. And we also found when we used methanol as a solvent, we could actually immobilize our nanoparticles onto the support without having to acidify it. So that was, that was actually quite helpful. You can remove one step in the synthesis procedure. And what we also found when we use methanol as a solvent, in the absence of acidification, you can see this sort of halo around one of the particles. This here is quite a large particle, but it does show the effect quite well. So there's something sort of quite significant that's adhered to the particle, and that's actually the capping agent. So what we're trying to do at the moment, and this is work that's not completed, but I just wanted to sort of show you what you're able to do. So actually with solid state NMR, you can actually assess that surfactant on the palladium nanoparticle surface. And what you can see is that actually when you use the methanol only sort of preparation route, you get a much larger quantity of surfactant absorbed onto the nanoparticle surface. So yeah, it sort of validates that sort of TM imagery. So going back to George's work, so again, we're looking at sort of furfural hydrogenation, and we compared his sort of methanol catalyst without acidification compared to the water series material. And actually, when you look at the inherent sort of conversion with the data here, you can see that on face value, the conversion of the methanol catalyst, it looks less. But when you look more sort of forensically, what you see is, well, actually, the conversion to, or the yield, the products we want is broadly similar, but what we're doing is we're suppressing the byproducts. You actually get a lot of ethers formed because you have an alcohol solvent, and of course you have sort of this sort of aldehyde functionality, so you can get ethers formed through different bronze and acid sites on a titania surface. And actually, by using our methanol route with PVA and without acidification, we suppress this unwanted byproduct. So we found a paper at the time which suggested that you could get bronze acid sort of catalysis via heterolytic hydrogen-hydrogen cleavage over palladium halide interactions. So we took George's samples to the BM28 beamline, that's the UK's beamline at Grenoble, Xmas, and we looked at the chlorine K edge. And here we have sort of, a collection of Zane's data. The sort of one response here is a reference material. And here we can see a very strong pre-edge feature just below 2825 electron volts, which is linked to non-local excitation between chlorine and palladium. And that's basically can be used as a proxy for the extent of chlorine-palladium interactions. So this hypothesis was to be correct. You would expect the one that has the higher intensity feature here to have more halide and potentially more bronze and acid sites, and you don't get that. So we sort of discounted that sort of idea. But when you do the TPR of these materials, what you can see is that when you look at the two different sort of series in sort of water, and so if you look in terms of the left-hand side of the screen for you, what you'll see is that sort of very large feature 3, that's for the catalyst prepared in water only, and that corresponds to hydrogen spillover onto the support. And you don't get such a large feature for that on the material prepared in methanol. So what we propose is that by having this sort of shroud or surfactant around your nanoparticle, it hinders hydrogen spillover, and in essence begins to switch off that bronze to acid pathway by those sites on the titania. And then interestingly, when you recycle this catalyst many times over and you end up degrading that surfactant, the selectivity profiles coalesce and you begin to get more of those ether byproducts. So this is a way that you can potentially learn how to control this step. On that sort of note about that spillover, there's other things you can do with that actually. And this is here is a collaboration with Alberto Villa at the University of Milan. And here we were looking at palladium nickel oxide for furfural hydrogenation. And what we saw, we compared palladium nickel oxide to palladium titania for this reaction. Again, very different selectivity profiles. The nickel oxide prefers the palladium on nickel oxide prefers tetrahydroferryl alcohol as a product, but palladium on titania prefers furfural alcohol as the product. And when we looked at the different surface sites through CO adsorption, through drifts, we didn't really see much of a difference. We couldn't rationalize these changes through the availability of different surface sites. And actually, what we postulated was that maybe the nickel oxide is doing the furfural activation, and maybe all palladium is doing is dissociating hydrogen and spilling over to the support. So then we made some catalysts of very different loading. We made 0.1% palladium on nickel oxide and 0.1% palladium on titania. So the 0.1% palladium on titania, we look back at the activities on the slide below. Titania was about 320 and 470 for the one on nickel oxide. You can see here, a tenfold decrease, which is what you'd expect if palladium provides your active site. But for the 0.1% palladium nickel oxide, we essentially maintain very similar activity. And this supports the view that actually what palladium is doing in the process is not the activation of furfural, but it's splitting the hydrogen, which goes on to the support. And that's where your hydrogenation sort of happens. So you can learn from your design strategy about how you can utilize these things that you see. Josie's raising her hand, so I'm guessing I've got 5 minutes to go, Josie.
No, you're more or less out of time now, Peter.
If I'm out of time, Richard, I will actually leave it there. Because that's fine. I will leave it there. I was going to talk about carbides and nitrides, but I think that's a good place to stop. So I'm happy to leave it there. So hopefully, just to sort of summarize, hopefully I sort of explain that it's really important to understand how you make your catalyst and how that imparts different properties to your materials, and also some information on the dynamic nature of catalysts during reaction. But yeah, happy to take questions now. And sorry for running over a little bit.
Well, thanks very much, Peter. Very interesting talk. I think we've time perhaps just for a couple of quick questions. Mike?
Many thanks, Peter. The lanthanum and manganese oxide for N2O decomposition, you stressed how important the defects are. Will it keep doing the reaction then? Is it only surface defects that activate the decomposition and then it stops? Did you try repeating the experiment?
That's a good point. We did try. So essentially, once you take it to higher temperature, you lose those surface defects. So yeah, you can't recycle it, Mike, in this sense. But we learned a little bit about how the catalysts were made and sort of, what was important. So you need to stabilize those defects if you want to have long-lasting activity at these lower temperatures. I hope that answers your question.
Thanks again.